Thank you, Rudolph. Hello, everybody. Thanks for coming, even though it's siesta time. So, Mike, I apologize for the complete change in my title and abstract. I'm still going to roughly talk about adaptation to environmental change, but I wanted to build in a bit more of the species interactions component that is the theme of this session. I want to thank, obviously, Rudolph for inviting me, but I also want to thank the organizers of this conference for what has been a splendid cultural and scientific experience. I've had an incredible time, seen some amazing talks, and met some great people. So I hope that continues on through the afternoon and into the banquet this evening. And before I go any further, I'd like to acknowledge the many people I've worked with over the years who are all responsible for my current intellectual thinking. And it's been a pleasure working with all these people. Many of them are students and postdocs in my lab, but also colleagues at other universities, and the funding agencies, who without that we wouldn't be doing much. So let me begin with the big picture. I believe that we as a field, as a domain of science, we need to be thinking not just about the deep imponderables of biodiversity, why it changes, how many species there are on planet Earth, and how those species interact to govern or to mediate the functioning of those ecosystems. But I believe it's our responsibility to connect those dynamics to human society, to sustainable development, to the economy. And we need to be doing a better job of connecting the fundamental principles of biodiversity and adaptation to environmental change, this top loop here, through to human society via our thinking on how ecosystems function and the benefits and the goods and the disservices that we face or we get from those ecosystems. This is imperative because this cycle has been accelerating over the last century. Biodiversity change is currently at an unprecedented rate, at least unprecedented in human history, and we have about a century to figure this out. Extinction rates are going to peak in the coming century. We recently reviewed this topic, and I think it's pretty clear now that biodiversity change has impacts on human well-being that go far beyond what we thought they were some 15, 20 years ago. Another important aspect of this figure is the spatial scale at which it occurs. Okay? Biodiversity change happens globally. We know that global extinction rates are massively elevated, but it's also happening regionally. We can see in these hotspots here, the redder the color, the greater the percent decline in mean species abundance across a range of vertebrates and invertebrates. And you can see where it's red, we've lost more than 90% of population abundance. What's key for me is that, yes, it's global, but it's regional, it's patchy. Right? The processes of biodiversity change happen. They're associated with human centers of population density, of course, and where we've massively transformed the landscape. So we have theory at the micro scale, right? Community ecology, population ecology tends to operate at scales of tens to hundreds of meters. And we have macroecology, which happens at the scales of entire continents. But I believe we're missing a fundamental missing link for me is the mesoscale approach. Can we predict the dynamics of biodiversity change and ecosystem function at the scale of a few hundreds to thousands of squared kilometers? That's where humans operate, and that's how we're going to manage this crisis. So I'm going to briefly mention and talk about a story that I'm currently embedded in right now, and I'm not going to be in the Mediterranean, unfortunately. I'm sorry about that, but I'm going to focus on where I've been living for the last decade, and that's in southern Quebec, in northeastern North America. So here's the story. About a year and a half ago, a member of the Quebec government called me up on the phone and said, "Andy, we'd like you to design an ecological network for the region of Greater Montreal." I said, "Sure, give me a day and I'll fire that up for you." And I realized when he'd done that, after talking for about half an hour about what it would take to do that, I realized that we didn't actually have the science in place to be able to roll out a designed ecological network for a region that is 200 by 200 kilometers squared. Okay? Montreal is sat right there. It's a city of 3.5 million people. There'll be 1.5 more million people in the next century. Many species are threatened in this region because it's been heavily urbanized and heavily converted for agriculture. but approximately 1% of this area is under some sort of protected status. We've got a climate that's warming. This region will be 2.6 degrees warmer, depending on the model you look at, by 2100. And there's roughly 10 to 5% of the remaining forest cover. So this region is massively transformed. It looks like this most of the time. It's not a terribly attractive, soybean fields with scattered patches of forest that are the hotspots for biodiversity in this region. And this region gives us lots of good things. We get, services like carbon sequestration, we get wood, we get agricultural products, and we get a little bit of pest regulation. But what the Quebec government wanted of me and of my team was to design a network that could optimize the benefits that we get from this landscape at the same time as maintaining biodiversity at this scale. And could we do that? What would it take to do that? What's the science we need to adapt the region to oncoming threats and to mitigate some of the darker sides of human impacts on this landscape? So I was thinking about something like this. We need a network that can identify key patches in the system, that this network would have properties that would allow organisms to move around this system. This network would be adaptive in the sense that the evolutionary processes required to make it adaptive would function over the next century. This system would have to be optimally connected, optimally from the point of view of the organisms involved, but also from the point of view of humans that manage the system. And we want it to be controllable in the sense that we can mediate the links between the patches and the patches themselves so that we can actually direct the whole system to desirable states, whatever those states might be. That's not a scientific question, that's a public policy issue, but at least something that can be designed to do that. And so what I think is we need a mesoscale theory. We need a theory that can couple our thinking from the micro and the macro and blend it to the regional. What is that theory? It's something I've been working on with many colleagues for the last 10 years. It's what we currently call meta-community ecology. This is the idea that the diversity we see around us at these regional scales is due to the fluxes of organisms, genes, and resources between local communities and their ecosystems. Okay? All communities are open. They're open to the fluxes of individuals and resources. And that's the key, one of the key features for me that defines the steady state diversity that we observe in a system and the transient changes, increasing or otherwise, if we modify the connectivity. So that requires, a theory requires some mathematics. And so we work on these, what can be quite ugly looking sets of differential equations that define the dynamics of the species and the resources they depend upon. And so in these systems, we define, in this particular system, it's a set of competitors that are competing for a limiting resource here, and the rate at which they consume that resource depends upon their traits. These traits govern how quickly they consume the resource, and the speed at which they consume the resource depends upon the quality of the environment, the state of the environment, little e, at that time. So if the environment changes, different species are taking up resources at different rates, and competition proceeds at different rates, and different species become winners or losers through time. And of course, we track how resources are being used up. So we do that for each species I over each patch J in our region, each community J in our region, and then we couple all these patches with dispersal, with movement. Our species move around this landscape. So here, we don't really need to go into the details, a very simple global dispersal model that propagules go up into the atmosphere and then they settle down again. That might work for some species, but it won't work for all species. So then what we do is we modify this bit here to represent a spatially explicit network of patches, where each patch, each community J has a set of coordinates in space, the Euclidean position in space. And then we can define things like complicated dispersal kernels, nonlinear rates of dispersal, etc., etc. So there I've given you a flavor of what the system might look like, and I'll show you in a second that we can add trophic levels, as many as we want. And because this model is very general, it's actually equally applicable to an asexually reproducing system, where here we're looking at clones of certain genotypes as opposed to species. So it's quite a general foundation. And the critical thing in this model, it turns out, is this parameter here, A, which in this particular version defines how connected the system is. It defines the degree of connectivity between patches and how organisms are moving around that landscape. So whether it's a white-tailed deer, a bumblebee, or a white-footed mouse, the rate at which they're moving around is key to determining many features of the meta-community. So here I'm always going to show you this dispersal rate on the x-axis on a log scale, and then various properties of the community and the meta-community. So you'll notice at the top here I'm showing you for 3 levels of dispersal, low, medium, and high. In the absence of dispersal, basically there's one species represented by a colored blob, and patches or different communities are represented by gray blobs. So when you have low levels of dispersal, you have a single dominant species in each patch. Alpha diversity is very low and gamma diversity is very high. When you turn the dial on the dispersal and you reconnect the system, here you'll see that now we've got much more local diversity. This connectivity is allowing species to colonize and occupy patches. And then we maintain at the same time gamma diversity. But now when we go to very, very high levels of dispersal, the system collapses back to one dominant species that covers the landscape in a monoculture. Now that's a fairly unrealistic limiting case, but what this means is that diversity in these systems is unimodal. There's a sweet spot where there's a maximum of local diversity and regional diversity at intermediate rates of connectivity. And what's very cool is that connectivity not only determines the number of species in a system, but also the properties of that system. So here we can look at the stability of productivity in the meta-community or at the mesoscale. And you can see that the variability is minimum. The system is most stable at intermediate or low to intermediate rates of dispersal. And the productivity of that system, the biomass produced per unit time, per unit space, is maximized at the same rates of dispersal. And these are the rates of dispersal that also happen to maintain diversity, local alpha diversity. So now we have a theory that gives us an underpinning explanation for not only patterns of diversity, but also the processes that those communities are doing. We've recently extended this to multiple functions and to more sophisticated types of dispersal processes, but the most significant extension from my point of view is that now we've added trophic diversity. So now we can modify the equations to take into account the degrees of generalism and specialism in the feeding interactions between those species. But most importantly, we can define the spatial scale of dispersal and the rate of dispersal. Okay, so now we can give species different properties of how they view the landscape. And it turns out that if you explore this parameter space, again, there's this intermediate optimum, this sort of Goldilocks effect, where we get the maximum complexity of our food web. So this is complexity as counted by the branches of our food web, okay, the number of minimum spanning trees that can characterize our trophic structure. But we can also in parallel, look at the accumulation of species richness in this space. And you'll see again that it's unimodal. Okay? And there's a sweet spot where we get maximum species richness, but also maximum trophic complexity or branching. I can't go too much further into the details of this model, but we're in the process of validating it. And one way we can validate it is to go to real data sets, real food web data sets, and use real network data sets as the input to the model and see how much of the natural structure of the trophic community, the food web, we can recover in the model by tuning the 2 parameters, the scale and the rate of dispersal. And so in this model, we did that. We took many food webs in the literature. There are lots now published, but here you see St. Mark's, which is a seagrass community in Florida. We've got a British grassland, Silwood Park grassland, and then we've got Chesapeake Bay. And I know you're not really supposed to look at all the details of this, but hopefully you can see that the peaks in food web size, or the number of species that our model can explain, the degree of food web branching, and the complexity of our system peak at different points along these axes of colonization rate and dispersal scale. So we get the biggest, or the best match between what we observe in the data sets by tuning these 2 parameters and nothing else. So that's good. Now our theory explains some realistic trophic structures. What about the dynamics of these systems? The dynamics of these systems are pretty complicated. And so we want to model the mesoscale, right? We want to study thousands of communities fluctuating in time. So here we take a model that in each location, so here there's a 65,000 food webs that are fluctuating through time, okay? We follow the dynamics. Here it is a very simple food web. We only have 4 species of a keystone predator and 2 consumers and their resource. And then we study their spatiotemporal fluctuations, because this is key to prediction. If we want to design this ecological network, we've got to be able to be sure that, like meteorologists, we can predict its dynamics. And the dynamics, are highly dependent on the type of processes that you model, and in particular, how dynamic the environment is. So on the left, what I'm showing you is the density of the predators, only the predators. And on the left, you'll see it's a very regular system, oscillating naturally, and that's because there's no environmental stochasticity. There are no environmental fluctuations. So the predator is just tracking the abundances of its prey. Now, when we add environmental fluctuations, you'll notice that occasionally we'll get these spikes, right? The system is not chaotic, actually, but it looks chaotic in a normal parlance, the use of the word chaos. It's highly dynamic. The predators outbreak in abundances sometimes, and what seem to be uncontrolled peaks. Then the system is quiescent, and we've got this thunderstorm of densities across the system. And so if we're trying to design an ecological network, whether we're in this case or in this case, the challenge is very, very different. And so now we're exploring what are the features of these mesoscale systems that allow us to predict what's going on the right as opposed to on the left. So I've just given you a sort of whistle-stop tour through the theory we've been working on over the last decade, and I think this is the theory, well, I know this is the theory we're going to apply and try to base everything on this idea of connectivity, the idea that all systems are open, they're non-equilibrium, and they're dynamic in space and time. Okay? And by dealing with that, we get for free an explanation for diversity at local and regional scales and the stability and functioning of the region. Now, of course, all good theory, theory's fine, but it's only useful if it's validated and tested. We do, like an engineer, I tend to shrink the problem down to the microscale, test theory in the laboratory first, before taking it out into the field and test driving it with real data. So we do a bunch of that in a lab. Alas, that's another talk. Well, I'll show you a bit of that in a bit. But I'm most interested in applying this in a complex field system. And so we've also, in parallel, for the last 10 years, been working on a system of a natural microcosm, so carpets of moss, that can be thought of as a natural microcosm of forest systems. So within these carpets of moss inhabit microarthropods, protists, bacteria, all sorts of mycorrhizae, all sorts of organisms. We have hundreds, literally thousands of species that interact at this tiny spatial scale, but they interact in complex ways, with food webs, with dynamics that look very much like communities with larger organisms. But what's interesting is that we can study the dynamics, the long-term dynamics over the scale of months to a few years. We can artificially warm these communities, fragment them, study the functioning of the ecosystems, the rates of decomposition, nitrogen fixation, et cetera, et cetera. So with this system, we've explored how changing the connectivity of that green landscape affects the standing diversity of the system. So if you fragment this landscape, you say you create 4 patches of moss, but you connect it with a viable corridor, right? You have this structural connectivity. What happens to the diversity of species in the system? So each dot here is a different species of microarthropod. It's inhabiting those moss patches. You'll notice that, so this is their density after fragmentation, and this is their density before fragmentation. So if the dots were lying on the one-to-one line, we'd say there'd be no change. You'll notice that, yes, fragmentation does cause a decline in almost every species in the presence of corridors, but look what happens when we break those corridors. If you break those corridors, then species are declining by several orders of magnitude in some cases. So here we've shown that the functional connectivity is assured by corridors and by the structural connectivity. But what about the function? The theory predicts that function will also benefit by corridors. And so here now we can track the fluctuations in the chemical composition of the system. We can look at the leachate and look at the rates of decomposition. And here I'm showing you how rates of decomposition, right, so carbon is being lost from the system, differs between these different landscapes. And what you'll notice is that over the period of about 300 days, up to day 150, there was no difference in our four landscapes. But over time, the connected and the disconnected systems diverged. So connectivity was maintaining natural, in inverted commas, levels of decomposition in our system. So we've confirmed that dual aspect of the theory that links structure to function. So take a break. I'm going to show you 5 minutes where now I'm going to shift gears. What I haven't really done is talk about the evolutionary resilience of these mesoscale systems. What is their capacity to adapt? Can they show genetic adaptation to the types of environmental change that I'm talking about? And can we design a network that maintains, that is evolutionarily resilient? So that's another new layer of research that we're tagging on top. What is the concept that we're interested in? It's called evolutionary rescue. This is the idea that a population that's declining and en route to extinction, of which there are many in this region and in the Montreal region, can reverse that decline, can win a race of demographic decline by going through rapid adaptation. And it turns out that there are some key features that allow a population to do that. It's better if the initial population size is big because the standing variation will be large, and the rates of the rate of the arrival of useful beneficial mutations will be greater. The stress severity can't be too big, otherwise the population will go to extinct too fast. We need to have a steady supply of beneficial mutations. And the theory makes all sorts of interesting predictions about how that might occur, but here is what it would look like in a simulation, right? You've got a population here that's declining. It's declining exponentially and recovering exponentially. But these lines here represent the arisal of beneficial mutations. So you're seeing genotypes that pop up, most of them go extinct, and then you get a rescue mutation that fixes and is responsible for the recovery of the population. And then you'll see there are a bunch more, and then there's another one that sweeps to dominance and displaces the initial rescue mutant. So these dynamics, these rescue dynamics, are well established in theory, but there's been almost no experimental tests of that, and it hasn't been extended to multiple species. So we've just recently defined the notion of community evolutionary rescue, where we look at the dual co-evolutionary processes between species and look to see whether we can rescue an entire community after a period of environmental change. Again, this theory needed testing. So what we needed was a situation where we could evaluate the probability of adaptation across thousands of populations, because the theory required that. So I could find no graduate student who was willing to culture thousands of populations for me, even with lots of money and alcohol and good salaries. So we had to employ a robot. And so we use these high throughput methods coupled to a fabulous genomic resource of baker's yeast or Saccharomyces cerevisiae, and we culture literally thousands of populations over many, many generations, exposed or not, to near lethal levels of stress. And in this case, it's salt stress. And what we studied was how often did our population go through this recovery, U-shaped recovery phase. And it turns out that it can do. There are certain situations that are important to allow that to happen. These are the stochastic trajectories in different wells, and you can see there are some populations that recover faster than others. You can see that some of them reach a lower minimum than others, and some just because of the stochastic process of evolution, increase faster than others. And what determines that, if you take the average, then you recover across many thousands of replicates, and you get an average that looks very much like the deterministic theory. Alas, I can't go into that, but we know the genetic basis, what are the genes that are involved in adaptation, because we've got this amazing genomic resource. The key predictor in this theory is population size. So the bigger the population, the more likely you are going to observe evolutionary rescue. Of course, that's a key feature that we can build into our ecological network, right? If we can maintain patches that maintain bigger population sizes and a better flow of useful genetic variation, then we should increase the probability of evolutionary rescue. So this is what we did. We asked the robot to control populations that range from billions of individuals down to millions, down to hundreds of thousands, down to a few tens of individuals, and we scored the probability of evolutionary rescue in the presence of high near-lethal salt. And what you see here is the result. This is the result for one of the experiments. We repeated the experiment under no salt and high salt. Okay, and what the fit here is from a theory that's been done by a group in Montpellier. So this is an independent fit from an independent model. So in the absence of salt, the populations do really well right down to a few tens of individuals. You do get some extinction at the bottom, but of course you do. You get this demographic extinction that happens even in a happy, well-adapted population. But when you have lots of salt in the system, now you're pushing the curve from left to right. Okay? Now you have to have a much larger population to observe a certain probability of evolutionary rescue. And in our case here, to have an equivalent of a 50% probability of evolutionary rescue, you'd have to have 500 to 700 individuals. All right, so if you want the probability that's equivalent to flicking a coin and saying, are we going to get rescue or not, you need hundreds of individuals in this particular species in this lab. If you want to have a 95% probability of adaptation, then you need to have thousands of individuals. So now we have this nice situation where we can begin to tune our system to optimize for the evolutionary rescue. Then, I'm going to cut this, I'm going to be very brief here. We took this to the next level. Of course, no population is isolated. It exists in a metapopulation. And so it was important to take into account the connectivity between our populations in our microwell plates. So we did that, and we modified the type of coupling between the populations and the rate of change or the rate of deterioration of environmental quality across our environmental ranges. And so then we asked the robot to take care of these metapopulations, to couple them at different rates, to degrade the environment at different rates, and then we scored how many evolutionary rescue events we had after 100 generations of adaptation to this degrading range. And I can summarize all these results in a single bar chart, which will be the last result from this talk. What you'll see is that there are several predictors of evolutionary rescue. So the number or the fraction of evolutionary rescue events depended in a constant environment where we don't change the salt. The type of dispersal is key. As you increase the rate, the scale of dispersal, if you go from local dispersal to global dispersal, you'll see that we can essentially double the fraction of evolutionary rescue events. So that's key. But when you have environmental change, now the history of selection overrides completely the effects of dispersal. And so now, if you have slow and fast environmental change, it doesn't matter how much dispersal you have, it's the fact that the populations have experienced salt stress in their history that determines the number of evolutionary rescue events. So population size, the rate of environmental change, and the type of migration in your network will determine the likelihood of evolutionary rescue. And we've just published a special issue on this, so I'm going to plug that. If you want to know more, there are 14 fantastic papers, some by speakers already at this conference, on this topic. So I've got about a minute left to come back to what the problem, the challenge that I got you thinking about at the beginning of the talk. How do we identify a network? So obviously I've been working on this for a couple of years, and this is my first attempt. This is my first attempt. Of course, there's a team of 20 researchers involved in this, so it's not mine, but it's the royal we. And here you can see what we've done is I've colored a forest patch. The darker the color of the forest patch, the higher the priority, the higher the conservation priority it should have from the point of view of its contribution to the dynamics of the entire region. So you'll see that these patches of forest here are of very high priority. And you'll notice there's a corridor that connects fauna and flora that are trying to leave the US and go to northern Canada, that passes to the north of the city. So that's the status quo today. That's what happens if you chose to lay the network down today. But unfortunately, land use change and climate change are ongoing. We model that. And this is what this network looks like in 50 years. You'll see that we've lost many of the key patches. The system is depauperate in key patches, and the system actually has difficulty maintaining diversity. So this is interesting, it's been fun, and I've spent much more time with politicians than I thought I would, and now this science is going to be incorporated into the Greenbelt for the city, the new Greenbelt that's been planned for the city of Montreal, and we're really quite excited about looking at the economic context of this and the political context to make this transfer, this science into policy. So a few conclusions. I hope I've convinced you that we need a theory at the mesoscale, that networks of networks can explain diversity and functioning at these regional scales, that we can build in evolutionary resilience. We know the principles of the theory of evolutionary rescue. Now we're extending that experimental program to more complex organisms with life cycles that are closer to the types of organisms we want to conserve. And I believe that we're at the limit, but we're now in a position where we can begin to do quite complex conservation science using state-of-the-art community ecology and biodiversity science. Thank you very much indeed.